All right, good morning, guys. You should be seeing this screen, the title screen here, and hopefully my mouse is moving. So we're going to talk a little bit about DNA replication and how it copies itself to either create new organisms or to fix anything that might need to be repaired. For example, making more bone cells if you break a bone, making more skin cells if you have a cut, things like that. Alrighty. Nope, just kidding. That's the wrong button. Okay. So the first thing, the definition of replication, that is the process of copying the DNA in a cell. Specifically, it's going to be in your nucleus. What you end up with at the end of replication is two identical copies of DNA. You hope, at least, providing that there's no mutation. One of the primary reasons, really, I guess it is the only reason, is to create another copy for cell division. Whether that's to create more cells for an embryo to grow, or if it is to repair a tissue that's already in use in a fully grown adult. So obviously with two copies at the end, one copy will end up in each cell. It's called, or DNA replication is generally called semi-conservative replication because you don't actually end up with a brand new piece of DNA. You actually end up with a half a piece of DNA in each molecule because you're going to have the DNA, which has two strands. It's going to split apart, and each strand is going to serve as the template for the new molecule. So each strand is going to have an original copy of the DNA, and it's also going to have a newly constructed piece of DNA. So that's why it's called semi-conservative, because it's not completely new. Like I said, generally it's to make new organisms, but it can also be used to repair DNA. Cancer cells actually can, this is usually where cancer cells have mutated. They've basically taken over the cell, and your cell doesn't know how to stop replicating. That's how you get the tumors, which is just uncontrolled cellular growth. So instead of turning off, these cells keep replicating, and then you end up with the masses, the small masses of tumors. So couple of pieces of vocabulary here. The first one is a template strand. This is going to be your original DNA strand. And then you're going to have a non-template strand, which is going to be your newly formed DNA strand. So, like I said earlier, it's called semi-conservative replication because each molecule of DNA is going to have one template strand and not one non-template strand. So here's the first step of replication. If you want to put like a little number 1 or something, I did not number these, but this would be the first step. Your DNA strand has to unwind. It's normally in that coiled compact shape because it is a very small nucleus in the grand scheme of things, and so we don't really have room for every piece of DNA to be completely unwound. So the entire strand doesn't actually unwind all at once. You're going to just unwind parts of it at a time because again, there's just not enough space inside of the nucleus. So you'll unwind one portion, it will get replicated, it will start winding back up, and then you will unwind a new portion. Reason why we want to unwind it is all of the enzymes that are going to be working on this replication need some space to get in there to match up the bases, to separate the bonds holding the DNA strand together, and so forth. So that's kind of why they have to unwind. The second step is that your DNA strands will unzip, meaning the DNA strands will separate. So there's a specific enzyme called helicase. You'll want to make sure you know what that enzyme is. Helicase's job is basically to break those hydrogen bonds between the complementary nitrogenous bases. That way you get two half strands, if you will, or two single strands. These are going to be your template strands. It says 2 chains of nucleotides, but don't forget there's also your deoxyribose and phosphate backbone on the side. Kind of a review. You had those in your worksheets the other day, but adenine and thymine have a double bond, and cytosine and guanine have a triple bond between them. Your third step, your extra nucleotides then are going to be attached to each template strand. They're going to be brought to the template strand by a protein called DNA polymerase. So its job is to bring in the extra nucleotides and form that non-template strand. There's actually another enzyme called RNA polymerase. It lays down a primer of DNA because the DNA polymerase just can't start building. It needs something to attach it to. RNA doesn't. So eventually that primer of RNA will come off of the non-template strand, but it's there kind of as a starter for the DNA polymerase to start building off of. There are also extra proteins called binding proteins that help support the original strand, your template strand, because instead of being more secure or more strong, I guess, with two strands of DNA, now you're a little less strong with only one strand. So it needs some additional support so it doesn't fall apart and break. And it gets that in the form of these binding proteins. So they're almost like scaffolding. Well, scaffolding is not the best word, but kind of like scaffolding to help hold up that template strand. So one thing you want to take note of, the DNA polymerase is going to be building from the 3' end to the 5' end. We talked about that with the DNA structure notes and the antiparallelism. So one strand is always going to run from the 3' to the 5' in reference to the deoxyribose sugars. And then the other strand is going to be running the opposite direction from the 5' to the 3'. So this is important. It doesn't matter which side of the DNA strand you're on, it's going to be built from the 3' to the 5' end. It's a good thing because then you don't have DNA polymerase proteins running into each other. If they got to build from whatever way they wanted to, then you might have some DNA polymerase proteins building from 3 to 5, and at the same time, you're going to have somebody else, another polymerase molecule, building from the 5 to the 3', and eventually they're going to run into each other, which would be bad. You want them to be building in the same direction. All right, next step. Since replication only occurs in that one direction, from the 3' end to the 5' end, one strand is always going to be leaning towards that replication fork. That replication fork, if you look at your diagram, that's basically right where the helicase is, quote unquote, unzipping that DNA strand. That's called the fork. So one DNA polymerase is going to be working towards the fork, following helicase. The other one's going to be working in the opposite direction. The one that's following the helicase is going to be called the leading strand. The other strand, the other template strand that's being worked on that's going away from the helicase, which I believe I've drawn it as the left template strand, that one's going to be called the lagging strand. Oh, and I have a typo. I just noticed that. So if you look at your diagram here, here's my typo. As helicase will be moving along the original strand, you're going to see that lagging strand is going to have pieces of DNA. It's not going to be one continuous strand of DNA. So that non-template strand on the left side is broken into pieces because it's going to have several DNA polymerase molecules working on it. So it's going to end up with these pieces. These pieces are called Okazaki fragments. The leading strand doesn't have any of these fragments because they're just being built in one continuous run. But the lagging strand will have these Okazaki fragments. Okay, and then here's where we're going to kind of stop for today, because that's a lot to process. But the next step is those RNA primers that were originally laid down on both the lagging strand and the leading strand. They're going to be taken off because we don't want them. We don't want DNA mixed in with RNA at the end. We want a full strand, continuous strand of DNA. And then another protein called DNA ligase, that's going to come back into that lagging strand, and it's going to go ahead and add in the blanks, if you will, and connect those Okazaki fragments. So it's going to bring in additional nucleotides to paste that lagging strand, or I guess to complete that lagging strand, to make it one continuous strand. So then you end up with two fully formed non-template strands. Additional proteins will bring the additional proteins will go ahead and connect those covalent bonds between the phosphate group and the deoxyribose sugar. So those are covalent bonds. And then you're also going to get those hydrogen bonds to reattach the strands together. So that template strand and the non-template strand. So this is kind of where we're gonna stop for the day. This is essentially the whole process of replication. We've got a few more things that happen after the two strands have actually been formed, but this kind of gives you an idea, the steps of the entire process, as well as those different proteins that are important: the helicase, the DNA and RNA polymerase, the binding proteins, DNA ligase, things like that. So just make sure that you're familiar with all of those proteins, what they do, and just the steps of replication. So that's kind of where we're going to stop today, and we'll pick up some extra stuff tomorrow. So if you have any questions, just let me know.